In this introductory video to Chapter 14, we'll be talking about recombinant DNA technology and the role of restriction enzymes and vectors in this type of technology. By the end of this video, you should be able to describe some important applications of DNA cloning and recombinant DNA technology. You should also be able to define the terms restriction enzyme and vector. And finally, you should be able to identify the function of each part of a vector. So first, if we're going to talk about recombinant DNA technology, we first need to understand what recombinant DNA is. So recombinant DNA is produced when a piece of foreign DNA is spliced together with another piece of DNA, such as this plasmid we can see here from this bacterium, to make novel DNA that can be put into a host organism. So basically we've recombined DNA from different organisms into a novel piece of DNA. So here we see an example of that. So we see a bacteria, and this bacteria has plasmid DNA. So we have the bacterial chromosome, and there's also this exogenous plasmid. So remember that bacteria have circular DNA. So what we can do is we can cut this plasmid open and insert a gene that we're interested in producing. So this might be a gene from a human cell that we're interested in studying, or it might encode a protein that we're interested in making so that we can study it. So here you can see now we have this recombinant DNA plasmid that we can put into a bacteria producing a recombinant bacteria. So what do we do with this recombinant bacterium? Well, the short answer is there are lots of things that we can do with it. So once we have these recombinant bacteria, we can use this bacteria as a DNA making factory to have lots and lots of copies of the gene. This may be used for basic research on this gene. So for example, we may want to insert this gene into a crop to create resistance to a pest or to a pesticide. Monsanto has actually used this technology to create Roundup resistant corn. So this has been very lucrative for Monsanto. In addition, this gene may produce some kind of enzyme that could be used to clean up toxic waste like this oil spill. There are also reasons that we may want to do basic research on this protein. For example, this could be a protein that's important medically to dissolve blood clots. It also could be a protein that we could give back to humans to treat conditions where that protein may be missing. So for example, you could have a child that stops producing human growth hormone too early, which leads to short stature. So we could give back that human growth hormone so that the child will grow to normal stature. So now, how do we make recombinant DNA? Well, one of the most important tools that scientists need to make this recombinant DNA are called restriction enzymes. So restriction enzymes are enzymes that cut the DNA backbone at palindromic sequences. So first, what is a palindromic sequence? You may be familiar with palindromes in the sense of madam, I'm Adam. So it's something that reads the same in the forward direction and in the backward direction. In the DNA, this means something a little bit different. So here you see, this is an example of a palindromic sequence. GAA-TTC, and then if we look at the opposite strand, GAA-TTC. So a palindromic sequence in the DNA is a sequence that reads the same in opposite directions on the template strand and the complementary strand. So restriction enzymes recognize these palindromic sites, and these palindromic sites are then cut by the restriction enzyme. So here you can see that this particular restriction enzyme cuts the sugar phosphate backbone between G and A on both strands. So because this is a palindromic sequence, when it cuts between that G and that A, it leaves an overhang called a sticky end. And the sticky end is basically a short sequence of DNA that's single-stranded. And this is called sticky because it will attach to any piece of DNA that has a matching sticky end. So for example here, this AATT sequence can hydrogen bond with a matching sequence that has a TTAA sticky end. So because of this property and because of these sticky ends, we can actually insert foreign pieces of DNA by cutting that DNA with the same restriction enzyme so that we have matching sticky ends. And this is a picture of this, so this is how we can make a recombinant plasmid. Here we have two pieces of DNA that are cut with the same restriction enzyme. Our plasmid is cut with this restriction enzyme EcoR1, and then we cut our foreign DNA with the same restriction enzyme. When that happens, we have matching sticky ends, which allow these two pieces of DNA to form hydrogen bonds with one another because the sticky ends are complementary, and then we can seal any remaining nicks in the sequence with our DNA ligase. Just like we learned about in the replication chapter, DNA ligase can seal those remaining nicks. So now I want to talk a little bit more about the plasmid. So what kind of plasmid would we want to insert our foreign DNA into? We generally use plasmids called vectors, and these are vectors that are used to shuttle foreign DNA into different host organisms. And these vectors have some really important parts. So first, all vectors have to have an origin of replication. And as you may remember from the DNA replication chapter, this origin is required for the plasmid to be replicated. Now this is really important if we want this bacteria to pass this plasmid on to its progeny. So the origin of replication allows for the plasmid to be replicated. So then we also have a sequence, we have a promoter. And as you probably remember from the transcription and translation chapter, the promoter allows the gene that you're inserting to be expressed. So it's where our RNA polymerase binds so that we can express a foreign gene in our bacterium. So next we have a multiple cloning site, and many times a multiple cloning site is either found before or after a tag, because we can use this foreign DNA to tag the protein so that we can visualize it. So this tag is frequently used to detect expression of your protein, and this could include a fluorescent protein like GFP, green fluorescent protein, or it could be some other type of tag as well. But these multiple cloning sites contain many potential restriction enzyme sites all in a row. So here you can see an example of a multiple cloning site. So we have sites for the restriction enzymes SPE1, KPN1, NCO1, HND3, and so on and so forth. And this is so that you have lots of options to insert your gene of interest. We have lots of options that we can cut that DNA with so that we can insert it. So finally, our last important piece of the vector is a selectable marker. This selectable marker allows us to identify host cells that have taken up our plasmid. So many times the selectable marker is in the form of antibiotic resistance in the case of bacteria, or sometimes in other organisms it can also be different nutritional resistance markers so that allow the cells to grow under specific nutritional conditions. But this is used to select those cells that actually took up your foreign DNA. Because you can imagine that if you transformed bacterial cells with this vector, perhaps out of 1,000 bacterial cells, only 10 cells actually picked up the vector. We can identify those cells by plating them on media containing ampicillin, because the bacteria that aren't carrying the plasmid are all going to die in the presence of ampicillin, and the only ones that will be resistant to ampicillin, meaning that they'll grow in the presence of ampicillin, will be those bacteria that actually took up your foreign DNA. So these vectors are used to express foreign DNA in host cells. And that's the end of this video.